I think you were at Oxford before, is that right?
I actually did start here. I started my transition from clinical clinician to data guy here in this building many years ago, 20 years ago now. So it's glad to be back. It's quick. As I just mentioned, actually, it's a long to be back. It's been 20 years since I was last in this building. I was actually hoping just to sit at the back and get a few ideas, but I think Caroline twisted my arm to say one or two things about how we think about causality in experimental genetics and where ML may or may not come into that. So, 3 slides. The first is a little on causality in experimental genetics. I'm going to assume a generalist audience here, so it's a little bit of a primer. And what I mean by experimental genetics is the natural counterpart to population genetics, right? So we are changing genes experimentally, whatever the technology, CRISPR, RNAi, to see what the effect is. And then I will give you a slide on one way to think about things. This is how we sometimes think about things at Ochre, and then a little on, because of course I promised ML, a little on where ML can come into this. But before we get on to that, we have, I might have just mentioned now, we have labs. Our main labs are Oxford, but we also have labs in Asia and the US. This is our lab in New York. This is actually a very unique site. We have 3 surgeons who operate this lab as an ICU where we keep human donor livers alive 24/7. It actually runs almost like a mini clinical trial unit. Now, the reason I bring this up is because, well, of course we love to brag about it, but it's very different. It's a very different notion, a very different idea from many other maybe biotechs or tech bios, as they like to call themselves, in our space. I'm talking about like the recursion bios of the world, those who like to do very big, high throughput genomics, perturbation style work, and often at a big compromise. And we'll get to that. So for us, and I think maybe the main story, the main theme I'd like you to hear in all of this is really it's this careful balance. If you're trying to model causality, it's this careful balance of causality, scale, and complexity. And complexity is a very big one in our field. So a quick primer on causality in experimental genetics. And it really does come down to these complexity trade-offs, so at least the way we like to think about things. Now, this is a gross oversimplification, but I tend to think of this world in 2 camps. Either it's pooled approaches, and what we mean by that is, for example, with single-cell CRISPR, we give each individual cell its own mutation of sorts, and there are various techniques around this. This is from the Satija lab in New York. They're a fantastic team. He studied with me many years ago, and what they do is CRISPR-based approaches, then extracting based on a certain feature using maybe FACS sorting and then single-cell profiling. The challenge with this is you can only ever study cell autonomous effects. And that's a shame for a company like ourselves. We're a liver company, you might have guessed from the previous slide, and we want to study very complex interactions. How, for example, gene perturbations in hepatocytes will affect immune cells, will affect stellate cells, will affect fibrosis, and ultimately cirrhosis. So that's why we need to lean into more arrayed-based approaches. And by that I mean is by plating cells, tissues, whatever you approach, in plates and studying. And it's in fact a fantastic paper from Lazar. This I think is in vitro, talking about how they can extract very complex features using imaging on cell-based approaches. The challenge you'll hear, the challenge you won't fully appreciate unless you're in the space, is that all of these companies only use cancer cell lines to do this. So it's a little bit like dressing a hamster in a tuxedo and calling it a human, I feel. So we really haven't solved the problem. And I think part of the challenge is accepting that you're never going to get all 3. You're never going to get causality, complexity, and scale. So how do we get that? By integrating data sets. And this is, again, it's one way, it's not the only way we do things, but this is one way we do things. What you're looking at is a UMAP here, but this is not single-cell sequencing. This is work we did with our friends at GSK, where we knocked out every expressed gene in the liver, multiple donors. This is all primary work, multiple donors, multiple disease states. I'm showing you one version of the truth here. And so every dot here, so this is a low dimension projection, every dot here is a gene that's been knocked out. Now you can do very clever things with this. You can directly model gene networks. And I know our colleagues at GSK are thinking long and hard about this to, for example, predict how a perturbation might have an effect on expression levels, which we can then map to biomarkers and disease states. What we like to do, one of our favorite techniques, is to map it to disease signatures. And here I can show you there are genes that we know are upregulating fibrosis and downregulating fibrosis. And we do that then with the natural counterpart. So we do a lot of single-cell sequencing across the globe. In Taiwan, we do a lot of non-MASH, so non-metabolic disease type sequencing, where we're really studying more viral hepatitis and its impact on the liver. This actually, I think, is a UMAP from a particular experiment where we took slices of human liver. We can also keep slices of human liver going for quite a while. And we can actually, by perturbing them with like TGFB and various things, we can actually induce fibrosis and temporarily single-cell sequence and extract those features. And from that, we get meta-signatures, and we look for those signatures and how they're being modulated in the causal data. Now, of course, there are lots of ifs, ands, or buts with that, but it's a good first approximation to get us going. So then, moving on to slide 3, and where does ML come into this? Well, that's discovery. Now we want to be able to validate it. And again, the notion here is we want to be as complex as quickly as possible. And we use many types of models. Top there, it's all primary human work. These are primary human co-culture models. The difference here is this is the only model that we're aware of in the world that can predict fibrotic effects from hepatocytes. And that's because we take hepatocytes from cirrhotic donors. So again, you can see where I'm going with this. These are very complex models. They don't scale as well. They're not cancer cell lines. We do the same with tissues. You're looking at fatty tissue. We've set up a clinical network in Taiwan where if you're having bariatric surgery under consent, we get a biopsy of your liver. We can turn each biopsy into about 150 slices. It's a lot, but it's not massively high throughput. And so what we do here is, and this is going back now to the data that we developed together with GSK, is we found that with these sort of models, with about 1,500, 2,000 genes, if you train the outcomes of that on the causal data, you can actually get very good predictions on sort of future work. So you don't have to then do brute force perturbations. And there on the far right, we've shown you, so there's predicting a well-known secreted biomarker of fibrosis. These were all genes that were completely untested, very strong prediction, very good prediction of the effect of those genes on pro-collagen. And then we can very quickly get on to testing the genes in very complex models. And so in effect, quickly move on to running the clinical trial before the clinical trial. So I'll leave it at that. Moral of the story is complexity, causality, scale. It's a finessed game. Thank you.